Okay, this is Lab 4, the lab we just did. There are two parts to Lab 4. Uh, One part is the more complex lab we did where we endeavored to use a colorimeter, which is this instrument that is shown. All right, Coleman, get it together. There we go. Which is the instrument shown over here, hooked up to the LabQuest. You had these vials with different suspensions of chloroplasts, and we measured the rate of photosynthesis, the amount of photosynthesis over time that occurred. We'll talk more about that. The second part of the lab was the less complex part, the paper chromatography part of the lab, where we separated the photosynthetic pigments from the magnolia leaf. So let's talk about these. The more complex lab, Lab 4B, was where you made the cuvettes, which are special little test tube-like structures that can be used to measure the amount of light that is passing through a fluid in the cuvette. We used the colorimeter to do that. You had a blank cuvette, which was simply used to calibrate your instrument, so it's really not part of the experiment. And then you had three other cuvettes. What you put in each of them was chloroplasts, and some water and some buffer, and also something called DPIP. Okay, you need to remember the function of DPIP. When photosynthesis happens, electrons jump onto DPIP and turn it from being blue to being clear. So the more photosynthesis occurs, the clearer the blue dye, the DPIP, the clearer it becomes, and the more light can pass through it. Okay, so in each of these vials, you had chloroplasts, some other solutions, and DPIP. Now, one vial you actually put, instead of live chloroplasts, healthy chloroplasts, you put boiled chloroplasts. In the other vial, you had regular chloroplasts, okay, so C-plasts and DPIP, and this also had DPIP. But in this vial, you covered it with aluminum foil, so it was in the dark, whereas this vial would have been in the light. Okay, so basically what we measured, the colorimeter measures how much light is absorbed. So if you have a cuvette that is very dark because it's full of the blue dye, when the light is attempted to pass through, the light gets absorbed by the blue dye and not very much light comes out on the other side. Whereas if the blue dye has been reduced, if it has had electrons from the photosynthetic light reactions fall onto it, the dye gets lighter and more of the light can pass through and not much light is absorbed. And so what should have happened is we should have seen some differences between light and dark chloroplasts and boiled versus unboiled chloroplasts. So we did. Our experiment turned out just fine. Let's see. I'm trying to see. Indeed, the chloroplasts that had not been boiled and had light showed the most photosynthesis. Those that were boiled did not do much photosynthesis because all their proteins had been denatured. Of course, those that were in the dark did not do much photosynthesis because they had no light to excite the light reactions and so on. You need to be able to identify things in this lab like an independent variable, which we are looking at here, the light and the dark. So amount of light is an independent variable. The temperature, boiling versus not boiling, that's an independent variable. And you need to know what your dependent variable was, which would be the light absorbance, which in turn gives us the amount of photosynthesis that is occurring. Okay, that is what that is directly telling us. Let's see. We did measure the amount of change in light absorbance over time. I think that's probably it. You should know what our control was for this experiment. In this case, normally a control is whatever is normal. In this case, the chloroplasts that were unboiled in the light was our control, and so we compare against that control. Okay, our graph where we measured, here we have time on this axis, and remember minutes, we always want to put our units. We had percent absorbance on this axis, and remember to put an appropriate scale for each. And our lines should have looked something like this. Okay, and they did look pretty much like this, more or less, okay, where these flat lines were the dark and the boiled. The flat lines were the dark and the boiled, and remember you would need a key to indicate what each line was. So these would have flatlined, they didn't do any photosynthesis, whereas this one would be the unboiled in the light, and what we see is that its absorbance was high. In other words, it was very blue, but over time the absorbance got less, meaning more light was getting through. And so we see that the photosynthesis must have been happening, and the dye was getting lighter, more clear over time. Okay? With regard to your pigment separation, you put the pigment on the chromatography paper, you put the end of it in the solvent, and of course you see these separations. You should be aware of the 4 pigments that we looked at. Chlorophyll B, which was the olive, chlorophyll A, the bright green, xanthophyll, and carotenoids. What you need to know is that chlorophyll A is the important photosynthetic pigment. That's the one in the reaction center. That's the one that's really contributing to the photosynthesis directly. The others are accessory pigments that help pass light energy to chlorophyll A, or they are what is called photoprotective, which means they are going to protect that chlorophyll A, that all-important chlorophyll A, from UV radiation. Okay? Do remember that DPIP was taking the place of NADPH, so DPIP would have shown up right here. This is an illustration of photosystem 2, photosystem 1, the electron transport chain. Our electrons would be moving in this direction, and instead of jumping on NADP+, they jumped on DPIP and turned it colorless. Okay. You might want to just remember what this was. This was a heat sink. You remember I had the aquarium up, and that was to absorb any heat created by the light, so that would not also be another variable. Remember things that were held constant, the amount of each liquid, the time they spent in the light, things of that nature. Well, actually, I guess the foil one didn't spend any time in the light, but the others did. The instruments you used would have been held constant, things like that. Remember what constants mean. Here is an essay question, and it is directly based on that lab with the colorimeter, and only they're using a spectrophotometer. It's a different instrument, does essentially the same thing. They tell you 3 samples that you had, boiled, foil wrap. This is exactly our lab. Construct and label a graph. Beg for this. You have a great opportunity for points as long as you don't make silly mistakes like not labeling an axis, or not scaling an axis, or not giving the units for an axis. Title the graph, the effect of independent on dependent variable. Identify the control, and remember, control and constant are different. Constants are things held the same. The control treatment was the standard of comparison. When in doubt, just give some constants. You might get points for them, and give your best shot at naming a control treatment, which we said was the normal quote-unquote standard of comparison. And then you're going to then it asks you basically to discuss the electrons and photosynthesis and so on. So that's that. There's the data they would have asked you to graph for that. All right, so that's Lab 4. We did that real recently. If you were not here to do it, please see me, and I will try to walk you through it or give you some data, let you see the instruments and so on. So that's that. Thanks for watching.